on synthetic RNA or IVT, then yes, you can confidently detect even with reasonable lower abundance, and you can provide editing levels, modification levels, and so on. But if you're, on the other hand, if you're doing nanopore sequencing and you have very few instances of a particular modification, it's not sufficient. You have not seen the complete compendium of 256 combinations, right? Assuming the 5-mer that I was talking. Then you cannot build a model. And yes, you could do probably a simplistic model for 2 or 3 locations that you've looked at, but that is not going to cascade or enable you to detect in other contexts, other sequence contexts. So that's the challenge with the downside. But yes, if you had already done synthetic RNA sequencing and all, I think that would work. But otherwise, yeah, there are positives and negatives. You could get a full-length molecule and start doing nicely, as Cynthia put, show different locations and which one would help you to do dwell times and which modification or context would can be captured using current levels and so on. And yeah, for instance, we have seen that when you have a charged modification, you see a strong perturbation of the current, right? It's and yeah, others have also reported something similar.
Yeah. So just to be clear, definitely, and I see the point, right? That of course, if you know where the modification, right, is or it was mapped, right, especially in the ribosomal RNA, a lot of them, in principle, especially in, we know where they should be, right? So doing a nanopore, especially if you see a difference, right, you can pick up on that, right? So I see the point that, of course, if you are, hunting or chasing some, novel modifications, right? So that can be obviously the challenge, right? Yeah. Yuri, go ahead, Cynthia.
Again, I think it depends on the situation, because in the ribosome, we really benefited from the fact that we knew where to look, we knew what we were looking for, and we had all the knockouts available, et cetera, et cetera. But our very first experiment with the ONT system was SARS-CoV-2 2.5 years ago. But we didn't have any clue what we were doing. Oh, Aaron knew what he was doing. I didn't know what he was doing. Anyway, we found these pseudouridines, and we're still pretty confident they are there in pretty decent occupancy. We reanalyzed that data with newer software recently, and so I think for something that's somewhat abundant and might go in at fairly high occupancy, like pseudouridine or M6A. Now, I'd say M6A is pretty good in the Drouch motif, and Eva Novoa has really shown us how well that can work. It doesn't work so well in other sequence contexts, but I think for dispersed viral RNA and mRNA, you can still find modifications, but you're going to have a hard time with anything that's completely unknown, unusual, and low abundance.
Sure.
Yeah, I am not doing nanopores. We're only doing normal sequencing, but we compared the data with Evanova, and we find the same sites. It's already good between nanopore data and RiboMed-seq, for example, but with some reasonable overlap. But I think we have to distinguish 2 questions, in fact. De novo mapping, which is quite different from a question of quantification. So de novo mapping, probably we have to use all possible methods. So you cannot trust one method and say, I am good for it and I have good signals, and maybe not reproducible with other one. So I always argue that we need orthogonal methods for de novo mapping, ribosomal RNA, viral RNA, mRNA, whatever. So another question is for quantification of modification level, which is very common question because snRNA modulation is known in all kinds of diseases. So precision of our methods for RiboMed-seq and hydroxy-seq is about 5% of molar ratio per site.
Wow.
So, and we made a bunch of technical replicates. So one or two positions are always more variable than others, but others are always below 5% of variation for technical replicates, of course, for the same RNA treated in parallel or triplicate, and we made a lot of it. So we consider that everything which is more than 5% for those sites is biologically relevant. This can be concluded. I don't know if any estimation exists for nanopore data, what would be accuracy or precision in this case.
Yeah. Oh, that is great. That actually is, first of all, it's great to know that you can depict like a 5% in difference. I think this is like one of the definitely big challenge, right? To really, define and quantify the stoichiometry. And again, I go back to my point that, even if you have a 1% difference, right, of a ribosome population in the cells, you can still make a huge impact. The point is how confident, right, we can be like to depict that kind of difference. So I'm not so much familiar with how much you can go, in quantify this difference with nanopore. If any of the, Sarath or Cynthia, how much you really can push there the envelope. So with the nanopore.
So I think that is modification dependent. This is something that Eva and others have also noticed. The accuracy with which you can predict, even if you use synthetic and best controls, that depends much on how much the modification tilts the signal. And when I say tilt the signal, it could be dwell time, it could be current, it could be many of the factors that could impact the signal. But yes, not all modifications are different. And in this context, I want to bring up something Cynthia nicely showed on adducts. We have seen that generally adducts help. When you have an adduct and you modify the protocol with an adduct for your modification, favorite modification, and then you run them down the pore, you will see a tilt in signal. But the challenge is when you add a bulky adduct, you start blocking the pores. And we have had some fun and not so fun stories on how much you can, how heavy a moiety and how charged a moiety you can start adding. So I haven't seen a lot of numbers on how much does the pore can tolerate at this point. But yes, I think that work that could actually help on what kind of, because this is important for structure mapping and also for even modification mapping in particular. Yes, I haven't seen much numbers, but yes, that's the exciting area, but it has its own set of challenges. If you start blocking a lot of pores, it's a useless flow cell. You can't even reuse, even if you wash and those kind of, yeah.
Like, for example, I don't think CMC adducts will go through. Have you tried that? That
was my question, Cynthia. Great you asked. I was actually saying, no, this is very important. This is very informative for all of us. So how big you can make it, right? The nucleotides, how much you can modify. Actually, I was asking, what about CMC and nanopore?
So I did tell you the dark secret for the structure mapping. We had a lot of reads thrown away. One, because they're fragmented. I told you the fragmented part. I didn't tell you why they were fragmented or what were the, because they're bulky enough, they start blocking, especially when you have multiple heavy 1M7 adducts in proximity, they just block the pore, and that's a dead pore, right? So you will have to run more. So yes, that's what. So there's coming back to the CMC adducts. The same story when you see them delineated. So this is where the single hit kinetics of shape sec comes into picture. So the way Julius Lux and others in the field have been doing, they've been trying to ensure that each molecule has just one modification that will, so that there is lesser problem with blocking the pores. But if you have multiple modifications in proximity, that can actually either block the pore just because it's heavy enough, or sometimes it results in structural alteration, local structural alteration of the RNA, and that could also, disorient the pore. So yeah, that's my 2 cents.
tRNA basically will have a more, there will be more problem at this point to do nanopore for tRNA just because they're more modified, maybe one close to each other.
I would think so. We haven't tried, so.
Okay.
Yeah, in our experience, if there's a lot of modifications piled up in a small space, sometimes you can get it to work, and other times it can be a software issue that, and I think Dr. Janga was just saying in his talk that there are these signals that are, quite difficult to read, and I think software will look at some of these and just say, bad, poor, throw it out, stop reading that, or something. I mean, I think it's not necessarily bad data. It's just that the window of tolerance has been exceeded, and then we need to work a little more closely with the people at ONT so they understand what kind of data we want to look for.
Yeah, good. Thank you.
I think for short tRNAs, well, Eva showed yesterday quite nice data for tRNAs, and I think they had nano CMC sec. I don't remember in the publication if they noticed that. I think it's abortive reads in this case, but
no.
If Eva is there, she can come here probably.
But I was looking if Eva, you say Eva would be the perfect actual person to be in the panel. I'm thinking she's online. There is another question from actually Adriana Dabrowska. It's like she's also in postdoc in my lab. It's for Yuri. That is fantastic precision. It is comparable among different sites, or it is sequence context dependent?
Well, I'm not sure that the problem is in the context of the site. I think it's hidden somewhere. The problem, we try to understand why some sites are more variable there than others, and it seems to be hidden in internal algorithms of Illumina machine, which can throw away some reads arbitrarily when first nucleotides are not good for passing filter. So, and this changes from one machine to another. We had a very bad surprises when we bought a new one, and our results were not comparable, in fact.
Okay.
This is something we have to take into account even for routine analysis. So if you're doing high-seq and then nova-seq and then next-seq, results may be different. And to my opinion, we looked on all bioinformatics. It's not bioinformatics, which is a problem, but already at the level of raw data, we have this variability, which is difficult to understand or to predict.
Yeah. So actually something similar. Sammy Jeffrey also, right. I'm wondering about choosing between an NGS method from Yuri or a nanopore method like Cynthia. When is one better or more useful than the other? Anyone want to comment?
Well, I think for exploring unknown RNAs, nanopores is much better. So we have to give credit for this method. But when we are coming to quantification and you know what you're looking for, probably ribomed-seq and hydroxy-seq are more appropriate. But depends again, if you're working on one particular mRNA, for the moment we don't have a targeted version of our protocols. We are working on it, but you have to sequence everything, and it's becoming costly, very costly. So cost for a nanopore would be much lower in this case.
Maybe I'll take a different approach. I think if your interest is to get the context of one or more modifications, and you want to do a cost-effective data generation, which can be useful for your lifetime, maybe it's a bit of an exaggeration, but you generate once nanopore data, the signal data can be used for multiple rounds. If the lab is interested broadly in single molecule mRNOmes, along with potential future utility of the data for mapping modifications, then I think, yes, doing at least one or few rounds of direct RNA sequencing is a good reference set, because it also provides you where modifications could be occurring on, because especially if it is a poorly characterized genome or transcriptome. So that's one way to look at it. The other context in which nanopore would be helpful is if you have, if you can generate synthetic RNAs with the modification, because not all modifications you can generate a synthetic RNA which is about 100 nucleotides long, because nanopore requires at least about 100 nucleotides long to run down the pore. If it is too short, it runs too quickly and you won't get much signal. There is that issue. So you need to have about 150 nucleotide long RNA polyallylated, where you could embed this where you can embed this modification and get capture of all the sequence context. Then yes, you can start using such kind of data for developing a model for employing it in any model system if it is a new modification of interest. But the problem is, for most of the modifications, we don't have synthetic companies generating such molecules, and they anyway get expensive for
Yeah, no, it's definitely it's a good point. Yeah, I mean, I guess definitely all these methods they have a pro and cons, of course, depend on always. I mean, I don't know, from my point of view, always the biological question that you're asking, right? I mean, definitely, one important aspect that's been, definitely discussed throughout the talks, also how the modification are, whatever, regulated or changes, right, between a different cell states, different stress, different, oncogenic transformations, right? So how you can, what is the best way, right, to analyze not so much the absolute value, the amount of the modification, but the difference between one, like one sample to the other, right? And there are many people that I know that are trying to also do this analysis in many biomedical samples, right, where obviously there is more heterogeneity in terms of cells, one thing that I've been always thinking about, right, how we can think about make a system to do in a single cell ribosomal RNA sequencing, right? So for example, we heard from Kate Kenmeyer this beautiful system to do methyl-6 at single cell level, right? So for example, for me in my lab, we've been always mainly interested in pseudouridine, right? Can we come up with something to monitor this at the single cell level? I don't know if you have any idea, or you wanna.
Maybe I can tell the challenges. I'm sure Cynthia would also agree. Our nanopore has this serious restriction on the quantity of RNA that is required. Okay. So it's about 500 nanograms that is required for nanopore sequencing, and that seriously limits a conversion of nanopore protocols at this point for single cell. But yes, there are some groups who have made some attempts in this direction. But you would get much lower throughput. You could start doing barcoding and pooling of samples and all those things, but they will have their own set of challenges in terms of how much coverage you would have per loci. I think that's because among all the RNA molecules that are present in a flow cell, only a small fraction dive through the pore. Mm-hmm. And that's the reason why you have you need a much larger sample input at this point. So the capture rate is low for nanopore sequencing. So we are not spending too much time on that optimization, but I'm sure there are folks who are working on improving that. But when that happens, yes, I would see both single molecule and single cell come together.
Yeah, no, that would be fascinating.
We with our methods, we are not at single cell resolution neither, because routinely we would need something like 10 nanogram of input material for ribomet-seq and 25 to hydroxy-seq. We can go a bit lower, but 5 nanogram seems to be a real limit which is not much for many applications, but not for single cell. For single cell, it's still a lot.
Yeah, no. Well, I mean, we had to think about also the future, but it will be fascinating, right? So to get something at this resolution. Another point maybe we could discuss, I mean, this is something that maybe was also related to your talk, Sarath. So, especially doing long read sequencing, right? And seeing how you can combine that approach to predict for RNA, obviously, structure, right? And how this can be, in combination, can combine with all this technology, right? To monitor how RNA modification, right, impact the dynamic, the RNA structure. I don't know if you do anything about this, or you want to comment on this point?
So again, the the complicated stuff is always under the rugs, right? So we tried to use DMS sec as well, and I think that was a bit more challenging. And to be fair, the DMS attempts that we did was more on synthetic data. It was not so controlled. The controls were not so clean, and there was so much noise. It was hard to delineate that. So it's important to have proper controls. It's also important to control when you are using these ships like DMS or other kinds of reagents, how much of modification are you performing? If you do extensive modification on and you want to perform direct RNA sequencing downstream, it would either block the pores or the signal is going to be masked, and you won't be able to have proper readout. So that's my take on the single molecule structure mapping. So if people are attempting to do structure mapping with this, you want to do different extents of modification, right? Exposure times for the modification on these unstructured regions before you start calibrating or saying that this is what I'm going to do, this much exposure. So, and for our, we were very clean, I would say, because the results show that we have tried to make sure that every molecule has one or very few modifications, and that actually is the reason why magically the structure starts looking, fantastically nice. And even when we did that, we did see other structures for this molecule. So this was the most dominant structure. We had other structures which are closer or some which were very far. So yes, there are challenges, and depending on what protocol you use, how much exposure you'll have. Yeah. So I don't know if that answers your question, Davide.
Yeah, no, it is. I mean, I understand obviously the challenge, right? And it's just, it's been fascinating, right? Because in the sense that obviously, even when we started to think about, what is the role, right? Ribosomal RNA modification, especially, I don't know, pseudouridine, no methylation, right? They can help, interactions, right? Between region of the RNA that are far apart, right? Then they can help to obviously increase the hydrogen bond, right? And make, of course, again, increasing structure of RNA or increasing that are still unknown. How modification of ribosomal RNA help proteins, right? It can be ribosomal proteins, can be a translation factor, right? To be more associated with ribosome or less, right? So at that to the point, we are in a moment where there are different techniques from different aspects of RNA biologists. And, the point here is how we can combine, right, in all together in one shot, right, to see, a bigger picture. So that's the major thing. But I can see obviously the major challenge. Yeah. Great. So, all right, Sammy, I'm wondering about the barriers for Cynthia to move to eukaryotic ribosomal RNA.
Yeah, so I talked a little bit about for E. coli, the fact that there's 7 operons, and there are 62 sequence variants among those 7 operons. So for yeast, there's 400 operons, and humans more. Also 400? Yeah, okay, so I have my data guy over here. He answered the questions for me.
No problem.
So, if you got 400 sequences for human ribosomes that are pretty similar, but somewhat different. I don't know how many sequence variants there are. How many times are those going to be CU sequence variants? So I go looking for pseudouridine, and I got to look really hard to make sure I'm not looking at a sequence variant. And yeah, I think it'll be cool. I think it'll be a lot of work.
Yeah.
Right?
Well, no, definitely that is an interesting point, because definitely there is in the context of eukaryotic mammalian ribosomal RNA, there is more once again, like, you know, different observation that there may be different variants, and how obviously these nucleotide variants of the RNA in the eukaryotic cells may also, you know, be they harbor different modification based on the variation. I think is still an open question, but it's interesting. Yeah. Yeah. Well, another point was, you know, how, for example, nanopore or NSG sequencing can be compared to, for example, mass spec, right? So we heard a lot of, you know, still capturing. And Shilong, I mean, Long, if you want to also join us, you're more than welcome to really, you know, combining, right, thinking about mass spec versus this other approach, right? I don't know if you guys, you want to, or Long, if you have any comment related to your technique versus nanopore or NSG.
Yeah, thank you, Davide. Yeah, definitely. I think, you know, now if we need different approaches, right? Really to verify those modifications. It's difficult for one single method. Like nanopore, of course, like Cynthia, she mentioned about, and they have advantage such in area like long race, and they can have a single, get a single molecule level. But I think that would be challenging for mass spectrometry, right?
Yeah.
I think mass spectrometry now can handle tRNA. It would be a stretch at this stage to get into longer RNA, like a ribosomal RNA, yeah, for this section. Unless we do like enzymatic fragmentation, right, to get to that range. So I think it will be like complementary. I think it also depends on what kind of question you are asking. So if you don't need to get to like a single base resolution, you may also look at the using mass spectrometry to look at like 100 base resolution modification. They can give you some information if you do parallel with nanopore or next generation sequencing that tells you about whether you have modification in this 100 non-RNA or that 100 non-RNA. For some people, maybe this is sufficient. So it really depends on, or this could be a first step.
I think this will be a major challenge, how to integrate now all this diverse data from standard RNA sequencing, from nanopores. This is also a question from the chat. I think this is the challenge because the data are different. Sometimes mass spec will be a great help to understand. We have a signal, but we don't know what it is. It may be just nucleotide diversity between different genes, and this is a challenge also. Mass spec can help to identify is it real modification or not, and so on. And all these methods, they have their limitations, but their advantages also. So their integration in some kind of database with treated data, not raw data. Raw data are not useless, but close to it, because one has to reanalyze them again and again, and this takes years now. So for complex data sets. So we need some kind of integration of treated data with all limitations author can indicate that this is certain, this is less, and this is uncertain at all. But I think the field requires this effort.
In fact, Scott Blanche also asked, can or not RiboSeq a potentially PacBio long reads data be productively integrated to achieve a more robust results?
They should be integrated, but how to do it?
Yeah.
We need the same sample analyzed in different labs, so mass spec, NGS, and so some kind of benchmarking. Also, what kind of data we have with different methods for the same sample. So, but
yeah.
They do offer different advantages, right? If you're doing PacBio, you're doing cDNA sequencing, there will be dropouts from the RT. You may not have complete end-to-end, but yes, you depending on which RT you're using, you could probably get pretty good coverage. While Nanopore has known problems at ends, like a poly A problem, and that has been improving. And then there's a 5' drop-off of signal, But yeah, so they complement, and when you're integrating with short-read RiboSeq or modification-specific protocols, I think it should give insights on where, what modifications, and how they impact translation or other outcomes. That's how I see. Yeah. Again, it's an open question. There will be new methods. I would foresee this multi-omic data would generate. Yeah.
Yeah. No, definitely. Sammy asked a question. I'm curious about ribosome diversity from Davide. How many diseases show altered small nuclear RNAs, and do these ribosomes have different ability to bind proteins, riboproteomes? Well, I'll answer quickly. This is a fascinating question, Sammy. It's a great question. It's something that we are really addressing in a different context. We are collaborating also with Maria Barna, where we are analyzing all the ribosome composition, for example, in different cancer type, also in different obviously tissues. Small nuclear RNA changes actually have been also observed, for example, not only in cancer, as I mentioned, but also in different cardiovascular disease or neurological disease. So I think it's fascinating and thinking about, as I said, how changing the small nucleolus really change the RNA modification, and then that's what we need. What is the best methods to analyze the ribosomal modification in a specific cells or specific tissues, and especially when the samples are precious, there are a few amount of cells, and what is the best way? Well, I think we are almost done, unless any of you want to make any final comments. This was actually a great discussion, a great session. So do you want to make any final remarks, or you're good to go?
My final remark is thanks so much to Sami and Kate. This has really been a lot of fun, very informative.
Yes, I agree with you, Cynthia. Thank you, Sami.
Yeah,
and
Thank you.
Thanks a lot.
All right. Very well. Great. This was a great discussion. I think, I at least learned a lot from you. So I hope that many other people do the same. So hopefully we didn't confuse anyone. But thanks again.
Sure. Good afternoon or